So now that we're all here, welcome to What's New in MPMI, the virtual seminar series of the MPMI Journal. I'm Jean Harris, I'm the editor-in-chief, and this has been a really fun project for us, started through the pandemic, and is a way that we can connect to authors and to our readers across the world. And that's been a really fun thing for us. And I think for authors and readers as well. So today, I have as my co-host, Juan Ramirez, who is an assistant features editor with MPMI. And you may have read some of his press releases. You may have read, he's been also very involved in getting first author bios on the MPMI Interactions website. So, so if you're a first author and you haven't gotten your name up there, maybe you should be contacting Juan and get a little bio up there. It's a great way for people to get to, know who you are. So,
sure.
Do you want to say anything, Juan? A little bit about what brings you here?
Yeah, well, first of all, welcome to everyone to this seminar. It's a great opportunity to be here. It's also a very interesting, very interesting paper by Professor Hewezi, and I hope you enjoy it all. And yeah, as Jean said, I do part of what I do as an editor, assistant feature editor at MPMI. Is I work on I write press releases for articles, but I also work with interactions, making bios of some authors, and to show a more personal side of the researchers. So because what we see, we don't know who is the person that is behind a paper, right? And especially if it's not a professor or like say renowned person. So it's to meet this writer or this scientist behind the paper, and it's a great space. So if you're interested and you're an author in MPMI, you can always contact me. Ashley, could you please share my email perhaps so people can be in touch with me? And yeah, welcome everyone.
Yeah, so Juan is a postdoc, and so if other postdocs are interested, we will soon be looking for a next cohort of assistant features editors with the hope that they will overlap for a year with our current cohort and learn more about this side of publishing and about science communication. So if you're interested in that, please let me know. We'll be posting that soon. So one or two more things before I get into our seminar. Today's speaker, Tarek Hewezi, and I'll tell you in a moment why I invited him and what really caught my eye about his paper. But first I want to say that we are going to be having closed captioning as an option. Of course, Zoom does that automatically, but it does need some scientific fine tuning. And one of the goals of this seminar series is to increase inclusion. That's why we have it freely available. That's why we're trying to encourage people from across the world to watch. But one aspect of inclusion is also making it more available for our readers, listeners who are hard of hearing or for whom English is not a first language. So if somebody would like to volunteer to help edit the transcript, please let me know, and we will very much gladly take this contribution, this volunteer contribution, to help make our work more accessible. Part of that accessibility, just as a reminder, we also have this wonderful MPMI podcast that we've been doing, and the last few episodes have been done by one of our other assistant features editors, Tess Diet, and they are featuring a speaker, Jennifer Lewis, who was one of our What's New in MPMI seminar speakers last year. And so she talks about a lot of things, including her science, the idea of back burner projects, and also her initiatives to diversify science. Let's see. One other note is that Morgan Carter, our third assistant features editor, is going to be live tweeting this seminar. So if you don't have time to watch the whole thing, you can always come back and follow her series of tweets. It will be coming out of the MPMI Journal Twitter account. Okay, I think we have a lot of other things going on, including a reviewer workshop. So if you wanted to know, like, how do you review a manuscript? I'm not involved in this, but 3 of our other editors-in-chief from other APS journals are going to be running that this afternoon in 3 hours, again, freely available for those of you who want to develop your skills in that way. It can be very helpful. Okay, I'm going to stop there, even though there's lots of other cool stuff going on, and turn it over to today's main event. I've invited Tarek Hewezi, who is a faculty member at the University of Tennessee in Knoxville. And I picked his paper because it really caught my eye. He's looking at this question of epigenetics. So how during plant microbe interactions, what are the different ways that the microbe tries to influence the plant and the plant tries to influence the microbe? And he's uncovered a really interesting story where the nematodes trigger silencing via epigenetics, via methylation, in particular of a microRNA gene. And I'll let him tell you the story, but it really reveals a lot about what's going on in terms of tolerance and susceptibility on the plant side of things. So very exciting. And he just told me earlier that this whole story started with a 2008 MPMI paper. So I'm excited to be able to offer you guys the next chapter. So Ashley is going to be typing in the chat how to the link to the paper. So because I know some of you will want a little more detail to dive into the details, and so those will be there. So I'm going to turn it over now to Tarek Hewezi. If you could please just say a few words about you and how you got here, and then your interesting work. Thank you.
Okay, thank you. First, I would like to thank you, Dr. Harsh, the editor-in-chief of Local Plant Microbial Interactions, for the invitation, for the introduction, and for organizing this very interesting series. So my name is Tarek Hewezi. I am associate professor of plant molecular biology in the Department of Plant Sciences at the University of Tennessee, Knoxville. And I work with plant microbial interactions since 2005. And today we're going to talk about the identification and functional characterization of differentially methylated microRNA gene during the susceptible and resistant interaction between soybean and soybean cyst nematode or heterodiglycinase. So I have a short introduction of few slides in which I am going to talk about DNA methylation pathways, how plant pathogen-induced DNA methylation change in the plant genome. Then I'm going to talk about the transcription and the maturation of microRNA gene. And the last slide in my introduction is about the life cycle of a cyst nematode. So as you know, in plant, methylated cytosine occur in 3 different sequence contexts. It's a CG, CHG, and CHH, where H donate any nucleotide except G or guanine. So after DNA replication and cell division, DNA methylation is maintained by what we call context-specific enzyme. For example, the CG methylation site is maintained by methyltransferase 1 and its homolog. VIM1, it's another enzyme, is contributed to the methylation of CG sites. CHG sites is maintained by the enzyme we call chromomethylase 3 or CMT3 and its homolog CMT2. CHH, it's in fact the majority of CHH sites are established de novo, but some sites are maintained through the activity of the enzyme we call domain rearranged methyltransferases 1, 2, and 3 through the RNA-directed DNA methylation pathway, which includes the REST component, the RNA-induced silencing complex containing the small interfering RNA of about 24 nucleotide lengths. In response to developmental signal, stress, pathogen infection, plant also can induce what we call de novo DNA methylation. De novo DNA methylation occur in all sequence contexts, CG, CHG, and CHH through the RNA-directed DNA methylation pathway, which involve DRM1, DRM2, DRM3, but DRM2 is the key enzyme in this process. As with any balance system, active DNA methylation is also important for controlling G-wide DNA methylation levels to make an intermittent balance between active DNA methylation and hypomethylation. So, and in all plants, there is many genes coding for demethylase. For example, in Arabidopsis, so far, 4 genes coding for demethylases have been discovered. So the first one include ROS1 or repressor of silencing 1, Demeter, Demeter like 2, and Demeter like 3. In the recent few years, several studies have shown that different plant pathogens, including bacteria, oomycetes, fungi, virus, as well as cyst nematode, can induce DNA methylation change in the host genome. This can be increased DNA methylation level. We call this hypermethylation, can be reduced DNA methylation level, and it can be hypomethylation. The change in DNA methylation can occur in protein coding genes, either in the transcript region, we call this gene body, or in the promoter, or even in the transposable element adjacent to genes. So what happened when phytopathogen-induced DNA methylation change in genes or transposable element? Most likely the change affect gene expression at genome level, inducing reprogramming of transcriptome or host transcriptome in response to pathogen infections. Despite it became very evident now that plant pathogen-induced DNA methylation change in gene and transposable element, information about DNA methylation in microRNA gene remained completely unknown before the publication of this paper. As you know, this microRNA is a small non-coding RNA molecule of about 21 nucleotides, which post-transcriptionally silence the target gene, which contain a complementary sequence, leading to post-transcriptional gene silencing. And this slide, I'm talking about the transcription and the maturation of microRNA. MicroRNA genes are transcribed by polymerase RNA polymerase 2 into a primary transcript. This primary transcript is about between 100 and 150 nucleotides. And one of the main characteristics of the primary transcript of microRNA is the formation of what we call a pen lip-like structure. Hair bend-like structure, stripes. This hair bend-like structure is recognized by dicer-like one and processed to the microRNA duplex. The function strand, the blue one, of the mature microRNA is loaded with Argonaute 1 into the RISC complex. We call this RNA-induced silencing complex, and then targeted to the target genes, which contain a complementary sequence. Once this complex bind to the binding site or the complementary sequence of the messenger RNA. So this trigger messenger RNA degradation or translational repression, leading to post-transcription gene silence. Then I'm going to talk about the life cycle, which is the last slide in my introduction. As you know, the cyst nematode are sedentary endoparasite of root system in many crop plants. These obligate parasites are in fact engaged in a very sophisticated and complex relationship with the host plant. These nematodes are capable of redifferentiate root cells into a permanent feeding site. We call this a syncytium, which is the nematode feed, grow, and develop to adult female to complete the life cycle. So the life cycle of the cyst nematode is not that complicated. It consists of 4 juvenile stages: eggs, and other adult male and females. So eggs, 4 juvenile stages, and adult male or female. So as you can see here, after hatching from eggs, the second stage juvenile penetrates the root system and migrate towards the vascular tissue, where the nematode select one compatible cell as initial feeding cell. Then after this initial selection, the nematode became sedentary and select about 100 to 200 cells surrounding the initial feeding cell and induce cell-to-cell fusion events, and then transform all of these cells to one large multinucleated feeding site. We call this a syncytium. From this feeding site, the nematode feed and develop to the third stage juveniles, or J3, fourth stage juvenile, or adult females. And then later on, the body of the nematode transform to a cyst or a lemon-like structure containing hundreds of eggs, which hatch under suitable conditions. So the objective of this study were to identify differentially methylated microRNA during the susceptible and resistant interaction between soybean cyst nematode, heteroderoglycinans, and soybeans. And the second objective is to determine the impact of DNA methylation on microRNA expression. We need to know if DNA methylation acts always as repressive epigenetic mark leading to gene downregulation, or DNA methylation can also act as active epigenetic mark leading to gene activation. In this case, this will be the microRNA. Let's see what we found. And the last objective, to functionally characterize a set of differentially methylated microRNA using overexpression of transgenic root systems. This is the 3 main objectives of this study. So before to study DNA methylation differences between the susceptible and resistant interactions, so we thought maybe it would be a good idea to develop a near isogenic line, one resistant and one susceptible. And in collaboration with my friend, Dr. Vince Patton in our department, we're able to develop a near isogenic line that differing in the SN-resistant gene RHG4, coding for serine hydroxymethyl transferases in chromosome 8. This isogenic line are highly homozygous and share more than 90% of their genetic materials. So these are obtained from individual F13 generation from a cross between a resistant parent, Fowler, and the susceptible parent, Anat. As you can see here, this nematode susceptibility assays from 2 independent experiments, one in red and the other one it's in blue, showing that the resistant type is in fact resistant, showing very low number of cysts per plant compared to the susceptible line. So I think the reason for that is to study the DNA methylation with minimum of genetic effect, because this material share more than 90% of this genetic of this genome. So the first step to study global DNA hemizylation between the susceptible and the cis interaction, what we generated whole genome bisulfate sequencing. The sequence we received is it's more than 20x coverage, and this obtained from SN infected roots and non-infected at 5 dBi from both lines. And we had 3 biological replicates. So a total number of 12 samples. So we have 2 lines, 1 resistant, 1 susceptible. We have infected and non-infected samples, that's 2. And we have 3 biological replicates. It's 2 by 2 by 3, it's 12. And then we determine differentially methylated cytosine and use this information to map differentially methylated cytosine to the promoter and the primary transcript region of all microRNA gene in the soybean genome. It's that already published or already known. It's about more than about 700 something, very close to 800 microRNA gene in soybean. So, and then we produce this graph. As you can see here, the infected samples, it's green. The control samples, it's red. This is a susceptible line. It's in the top panel. And this is susceptible line in the top. And the resistant line, it's in the bottom. What you can see here, this is the global level of DNA methylation, or the absolute level of DNA methylation. You can see here what? The green lines, it's a little bit higher compared to the red. So this is mean there is increase of DNA methylation level in the infected samples compared to the control. The control is red, infected, it's green. So, and we saw this trend of increased DNA methylation in all sequence contexts, CG, CHG, and CHH. We saw the same trend of increasing global DNA methylation, the green line, also in the resistant line. And CG, CHG, but under CHH is not really very clear. So the conclusion is that DNA methylation seems, the infection of a soybean cyst nematode seems to induce global increase in DNA methylation in both lines. But when you look at this graph like that, you will not be able to see if this increase is statistically significantly different or not. It's just a trend of increase. To answer this question, we did Boxplot analysis to determine if this difference in DNA methylation level is significantly different or not. As you can hear in the susceptible line, the near isogenic line, the susceptible line, so the infected samples, that's green, the control is red. As you can see here in all sequence contexts, CG, CHG, and CHH, there is a significant increase in global DNA methylation in infected samples compared to non-infected control samples. This is for the susceptible line. We saw the same trend in the resistant as well, but the CHH, the difference was not statistically significant. So our conclusion from this analysis is that soybean cyst nematode infection induced significant increase in global DNA methylation over microRNA gene in both susceptible and the resistant lines. So what was the next step? The next step is to identify statistically significantly differentially methylated microRNA in both lines under infected conditions. To do that, we divided the promoter of microRNA, which we consider about 2 kilobase, to 500 non-overlapping windows, 500 base pair, non-overlapping windows. And we use this region or this window to compare DNA methylation between infected and non-infected samples in both lines, susceptible and resistant line. We also compare the primary transcript regions of this microRNA between infected and non-infected samples in both isogenic lines. We also use a methylation level of at least 25% with low discovery rate or forced discovery rate or adjusted B value of 1%. Using this criteria, we are able to identify 82 differentially methylated microRNA in the susceptible line. This number is significantly reduced in the resistant one. We are able to identify only 16 differentially methylated microRNA. So we have 82 versus 16. As you can see here in this heat map, this microRNA can be hypermethylated, it's red, or hypomethylated, showing reduced DNA methylation level in green. And this in all different sequence contexts. And also you can realize that many of them, about half of them, are in CHG sequence context. In the near isogenic line, the resistant, we identified only 16. And as you can see here, some are hypomethylated and the other are hypomethylated. Also, if you look very carefully at this heat map, you can find that there is 4 microRNA, labeled them with blue color. In fact, these 4 microRNA are common between the 2 sets of differentially methylated microRNA in the susceptible and the resistant. But they are showing the opposite methylation pattern. So if they are hypomethylated in the susceptible line, they are hypermethylated in the resistant. So they are showing the opposite methylation pattern in the 2 lines, and we thought this is very interesting. This set of 4 microRNA for further functional validation using transgenic repeat system. And I will talk this in a few minutes. So we next examine the methylation direction, context, and location of the differentially methylated microRNA. I mentioned a few seconds ago, in the susceptible line, we identified 82 differentially methylated microRNA, 34 are hypermethylated, 53 hypomethylated, and 5 showing both hypo and hypermethylation. How this happened? In this 5 microRNA, we identified some differentially methylated region with increased DNA methylation level, and another region with reduced DNA methylation. This is what we call this hypermethylated microRNA. In the resistant line, we identified 16 differentially methylated microRNA, 10 are hypermethylated, and 6 were hypomethylated. This DNA methylation, in fact, found in the promoter, as well as in the primer transcript region, but to a much lesser extent. And also this methylation found in half of them in CHG, this is the green color, and CHH about 1/4 in CHH and CG. So from this analysis, we have 3 main conclusions. First, that soybean system method induce hyper and hypomethylation in microRNA. This hyper and hypomethylation occur in the promoter and the primer transcript region, mainly in non-CG context. So we next examine the promoter of differentially methylated microRNA identified in the resistant and susceptible line for the presence of transposable element, to see if the presence of transposable element in the promoter of this microRNA gene may increase the susceptibility or the tendency of the microRNA for DNA methylation change. As you can see here, we found 37 of the 82 differentially methylated microRNA in the susceptible line contain transposable element in the microRNA promoter. Also, we found 5 of the 16 differentially methylated microRNA identified in the resistant line contain transposable element in their promoter. And this transposable element belongs to GEP-C and Cobia family of transcription factors. So we can conclude that the presence of transposable element in the microRNA promoter may be enhanced microRNA vulnerability to DNA methylation change. in response to pneumatic infection, but most likely is not the only factor, determining the methylation of the microRNA. Then, to gain insight into the biological processes in which this microRNA function. So we determine the target of this microRNA. We're able to identify 332 targets for the 82 differentially methylated microRNA identified in the susceptible line, and 77 targets for the 16 differentially methylated microRNA identified in the resistant line. And then we did GO-term analysis. And it's very interesting to see that the identified GO-term or they are involved or related to cell differentiation, organ morphogenesis, and the cell fate and the specification, as you can see here. So it seems like this is very interesting. It's, and when we found this result, okay, this makes perfect sense. Why? Because this process is very important for syncytium differentiation, formation, and development. So it's not random. It's not, soybean system method. It does not induce DNA methylation change randomly. It's targeted. It's target microRNA, in fact, that really was function related to soybean cis nematode interaction. So once we determined, once we did that, so we want to see if DNA methylation impact the expression of the microRNA in which. So we studied the expression of 4 microRNA that showing opposite DNA methylation between the susceptible and the resistant line. This is microRNA 5032, 5043, 1520P, 5770AB. So as you can see here, these 4 microRNA showed the opposite DNA methylation pattern between the near isogenic lines, the susceptible and the resistant lines. So, and this our analysis will be focused on this on these 4 microRNA. So, in fact, we studied the expression of the microRNA at 3 different time points. At 3 dpi, 3 days post-infection, 5 dpi, and 10 dpi in both lines. As you can see here, this is a microRNA. This is the expression of these 4 microRNA are oppositely regulated in the susceptible and the resistant line. Okay, just look at the 5 dpi. You found all of these 4 microRNA are down-regulated, but in fact, in the resistant, in the susceptible line, 2 of them are up-regulated. Also look at 3 dpi. You found 3 are up-regulated in the susceptible line, but none of these 3 are down-regulated in the resistant one, showing no change in the expression. So when we look at that, we see that the differentially methylated microRNA, these 4 microRNA showing the opposite methylation pattern between the resistant and susceptible, showing the opposite regulation between the resistant and susceptible. Also, I would like you to look at the gray bar here. It's the microRNA 5032. What is this? This is microRNA. It's hypomethylated in the susceptible line and hypermethylated in the resistant. So it makes sense. We found this microRNA. It's downregulated in the resistant line, which is consistent with the function of DNA methylation as a repressive epigenetic mark. That makes perfect sense. But also I would like you to look at the green bar. This is a microRNA 1520P. This is microRNA. It's hypermethylated in the susceptible lines, but showed upregulation. This microRNA also is hypomethylated in the resistant line, but showing downregulation. So DNA methylation can function not only as a repressive epigenetic mark, but can act as active epigenetic mark. Then we next quantifying the expression of the target gene of these 4 microRNA. So we determine 6 target gene or 6 confirmed target gene of these 4 microRNA, and we study the expression of this 6 target at the same time point at the same tissue samples. And also you can see here, this is the targets, the 6 targets of this 4 microRNA, also showing the opposite regulation pattern. It's very obvious at 5 dpi. All of them, 5 of the 6 genes are downregulated at 5 dpi, but 3 of them are upregulated. Also, you can see here at 3 dpi. So there's some genes are upregulated here, but not in the susceptible line. So we can see also, similar to the microRNA, their target gene showing the opposite regulation in the resistant and the susceptible line, at least at 1 time points. So it seems like the hemisization control or contribute to the regulation of the microRNA gene and indirectly their targets. Then our third objective of this study is to study or functionally validate the impact of a set of differentially methylated microRNA in the compatibility of the interaction between soybean and soybean system adult. So, and then, okay, this 4 differentially methylated microRNA showing the opposite pattern of DNA methylation between the resistant and susceptible line could be a very good, candidate to start as a function validation experiment. And I would like, to share with you a brief description of the method we use to generate transgenic hairy root in my lab. So as you can see here, the 5-day-old seedling were inoculated with agrobacterium. And then after 4 to 5 weeks, you can see here, this is the transgenic hairy root were nicely developed. We cut off the main roots, and then we select the transgenic hairy root with the aid of GFP, which our binary vector container GFP to facilitate the selections. So we selected the transgenic hairy root. So the composite plants containing at least 3 transgenic hairy roots were selected and planted in trays containing sand, then inoculated with about 3,000 eggs of soybean systematode. And in this case, it will be RES3 or HG type 0. After 5 weeks of inoculation, we extract the cyst from each plant individually and we count them under microscope. And we use these counts to determine plant susceptibility. So this is the cassette we use to generate our transgenic hairy root lines. So we overexpress this 4 microRNA, the primary transcript of this 4 microRNA, under the glycine max ubiquitin promoter and Rubensco small subunit terminator. And also this cassette contains GFP and expressed under the 35S promoter and NOS terminator. This is GFP normally we use to select the transgenic hairy roots. So we quantify the expression of this microRNA in few samples of the transgenic hairy root. As expected, we can see an increase of the expression of this 4 microRNA in the transgenic hairy root samples. And we also found the target gene of this microRNA, which shares the same color. 6 confirmed target was downregulated. This is consistent with the role of microRNA in post-transcription gene silencing of the target gene. So we have this data, in fact, validate the functionality of the construct used to generate the transgenic heredity. So then, after inoculating this plant, this line, we have at least 3 biological replicates, each biological with about at least 5 plants. We inoculated them with about 3,000 eggs. And after 5 weeks of inoculation, we collected the cysts and we count them under microscope. And we use this count to determine plant susceptibility. And we also generated transgenic plant expressing the empty vector as a control. And in the next slide, we share with you the results of the overexpression lines. So as you can see here, this is the empty vector expressed in a susceptible line, which shown about very close to 300 cysts per plant. Transgenic heliroot or composite plant expressing the microRNA 1520B and 5043 showed about 25% reduction in the number of cysts per plant. But this reduction was not statistically significant. Look at that. The composite plant expressing the microRNA 5770B. This plant showed more than 50% reduction in the number of cysts per plant. This is very interesting. But what is remarkable, it's the overexpression of microRNA 5032. Look at that. So the average number of cysts per plant, it's less than 10. It's about 7 or something, compared to about 300 cysts found in the empty vector or control plant. So this is microRNA able to convert alone the susceptible line to be very highly resistant. So what is the function of how this microRNA mediates this resistance? So microRNA 5032, it's in fact target 2 genes encoding glycoside hydrolases. And in fact, this enzyme involved in breakdown of sucrose and other complex carbohydrate molecule into small molecules such as glucose. And glucose is considered the major source of carbohydrate input in the sensation. Imagine if you are down-regulating this enzyme or inhibiting the activity of the enzyme. This, in fact, will affect the concentration of glucose in the sensation. And this will affect the sugar pool and the feeding site, and leading to nematode starvation and developmental arrest, and hence plant resistance. This microRNA 5770 also target 2 amine oxidases, which play a key role in polyamine biosynthesis. And maybe downregulation of this amine oxidase impacted polyamine concentration in the nematode feeding site. And we know that nematode effector protein 1006 target polyamine pathways. So this is it's in connection with our previous discovery that the polyamine, its key or polyamine pathway play a key role in a plant nematode interaction, and this may be connected to that. With that, I reach my conclusion. So we can say based on our analysis that the DNA methylation change are more dynamic in the susceptible line or the susceptible interaction compared to the resistant interaction. So as we are able to identify 82 differentially methylated microRNA compared to only 16 in the resistant line. We also can say that the DNA methylation occurred in the microRNA promoter and to much lesser extent in the primary transcript region, mainly in non-CG context, CHG and CHH. Presence of transposable element from Kobe and Gypsy family in the promoter of microRNA may enhance the susceptibility of microRNA to DNA methylation change upon SN infections. Also, we found that DNA methylation can act as repressive epigenetic marker. This is well known, but as well as an active epigenetic marker that can lead to activation of microRNA expression. We found that overexpression of two microRNA 5770B and microRNA 5032 significantly enhanced plant resistance to soybean systematic RES3 or HG type 0. The function of microRNA 5770 in the synthesis in soybean resistance may be associated with a modulating polyamine concentration in the feeding site, while the function of microRNA 5032 in significantly enhanced plant resistance, converting the susceptible line to be completely resistant, may be associated with depletion of sugar pore concentration in the synthesis, which affect nematode development. With that, I would like to thank all my lab members involved in this work, and also my collaborator, Dr. Vance Pantalon, for generating the near isogenic line used in this study. I would like also to thank funding support from Tennessee Soybean Promotion Board and from the Institute of Agriculture at the University of Tennessee. With that, thank you for your attention, and I'm happy to answer any question if I can. Thank you.
Thank you, Tarek, for this excellent talk. Very interesting. We already have lots of questions showing up. If you could unshare your screen, then we will move to the question period. So I'm going to start with reading off some of the questions, and Juan is going to take the next one. So Pawan Kumar asks, soybean is a polyploid crop. How many numbers of copies of these 4 microRNAs are present in soybean?
Okay, that's a very good question. So these 4 microRNA, one of them, it's there is more than 16 copies. Okay, but microRNA 5032 only 1 copy. MicroRNA 5032 is only 1 copy, and we are studying it because it seems like it's we have a very active project about this microRNA because it seems like it's translocated from the chloroplast genome.
Very interesting.
Yeah, like biologically, it's interesting to think why would one be 15 copies while the other one is 1 copy, and I wonder do you know how between different like varieties of soy, how like conserved are these microRNAs? Is it known or not really?
Yeah, when we look at this microRNA sequence between resistant susceptible line in different varieties, it's very conserved.
Okay, and the number of copies as well, or only...
It's the copy number exactly the same. Only 1 copy of this microRNA 5032.
Okay, interesting.
And we're trying to see from where it comes. So, and found it seems like from the chloroplast, and maybe acquired the new regularity functions. Okay. So we have a big project about it right now. So
yeah, it would be interesting to see, for instance, in other leguminous, to see the presence of
I'm doing it now. Yes. Yeah.
If it's related to resistance as well. Okay. So I'll go to the next question. They say, okay, hi, excellent work. In the transgenic heavy root experiment, is it a transient overexpression? If yes, how long do you observe the expression of the microRNA?
Okay, so when we have overexpression, so when we collect the roots after 4 or 5 weeks, so we sometimes we look at the overexpression and see the root is still expressing the JFP. So it seems like you can have the be active in the transgene hairy root for long times. Okay, and most important, it's very important. So you have this GFP or this microRNA expressing during the early stage of infection. Okay, because everything is happening in the first couple of days. You are inoculating the method. If they are expressing there, everything is done. Okay, so you don't worry about 5 to 6 weeks later on, unless you are doing different type of analysis of different type of functional validations. Okay, so you inoculate them. Next 2-3 days, the nematode interaction with the host plant penetrate in the first, maybe 2 days, 48 hours. So it still be there, right?
Yes, interesting. And our next question also looks at timing here. It says, nice, is by Dirk Schenke. It says, nice data, but methylation was investigated only at 5 dpi. So now that you have your candidates, will you be checking DNA methylation at these loci at all 3 time points using ME-DIP qPCR to clearly correlate DNA modification with gene expression?
This is a good question, but our focus was that at this time points. Okay, so we studied gene expression, but I agree there is no data about DNA methylation at different time points. But our focus was only this time points because we think this is what the majority of cellular change or it occurred. Maybe later on when we did in Arabidopsis, we see DNA methylation also, you can see it at later time points, but it's completely reduced. It's significantly reduced. So based on previous analysis, we know that we have early stage, like 3 dpi, 5 dpi, 10 dpi. We determined that the 5 dpi is it's the best time points where all biological processes, the molecular function associated with plant nematode action occur at this time. This is the peak.
Yeah, I have a question related to this, and it's, for instance, I was thinking when you say okay, we see that there is some loci, right, in which there is an increase in the DNA methylation levels, but we also see an increase in the level of expression. So it's not always that it has a repressive effect.
Yeah.
But I was thinking, do you know, for instance, if these loci are decorated with other histone modifications? Because we know that, for instance, DNA methylation and H3K27M3 there is also a repressive mark. They never go together, right? So, for instance, it could be some other epigenetic mechanism that is repressing this.
Okay, this is a very good question. So, so everyone knows that DNA methylation acts as a repressive epigenetic mark.
Yes.
Always, early papers published in Nature Science, PNAS, and everyone, DNA methylation, down-regulate gene expression. But in fact, when we can go deeper and deeper, DNA methylation is not always associated with gene downregulation. And we confirmed that in many cases, in soybean, in Arabidopsis, and tomatoes. What happens? This is my explanation. So what you have some DNA methylation can interfere with the binding site or the cis element of transcriptional repressor. In this case, you prevent the repressor from down-regulating gene expression, right? So in this case, DNA methylation activates gene expression because interfering not with transcription activator, it's interfering with the binding affinity of transcriptional repressor. That's one explanation. Second, sometimes maybe you have a transposable element located next to protein coding genes, okay? And sometimes many of them even within the gene. So if you have DNA methylation in the transposable element or in the promoter for genes, what happens? This DNA methylation prevents leaking or cryptic transcripts generated from the transposable element, right? And the correct transcript interfere with the correct expression of nearby genes. By repressing or preventing the leaking transcript from the transposable element, they can lead to increase in the transcription activity of the nearby genes. Okay, because you are preventing, in fact, the transposable element from interfering with the expression of nearby genes. This is the second explanation. Third explanation. So this is discovered, a few years ago in the human transcription factor. We have some transcription factor have very high affinity for methylated cis element. So in fact, they bind only to methylated cis element. And maybe some mechanism like that in plant. We didn't discover this yet, but maybe exists. So maybe you have transcription factor that professionally or high affinity for cis element or binding motifs that are differentially methylated or highly methylated. This can lead to gene expression change.
Okay. Great. So that was fast.
So that's the explanation. I can provide you another explanation that you can say, okay, to convince you that DNA methylation can act as, active epigenetic mark and can increase the expression of protein coding gene and microRNA gene as well.
But do you know that in general? Sorry.
I was just going to say, I love how our understanding of methylation is becoming so much more nuanced than sort of initially we had thought. So this is very fun to hear this discussion. Go ahead, Juan.
Yeah, do you know about if these microRNA loci, they can control as well by histone modifications, or in general only DNA methylation, or like are they in general enriched with the nucleosomes, these loci as well, or not?
No, say again. I didn't get it. So what's your question again? What?
Like, do you know if the loci that encode for these microRNAs,
yes,
they are also decorated with histone modifications? Are they enriched with nucleosomes, or in general they are just naked DNA?
No, we didn't look at it. We didn't look at it.
Okay.
Lots of good future directions.
Yeah.
So I'm going to jump to the next question by Dr. Tapan Kumar Mohanta, who says, we all know in stress conditions, nucleic acid undergoes methylation. It's one of the main molecular defense mechanisms. How do overall methylation events in the cell get increased, or is it only concentrated in microRNAs? And where do the methyl groups come from and participate in the methylation process? So these are kind of two questions. The first is more about general levels of methylation changed, and the second question is, where does the methyl group come from?
Okay, the methyl group this is an easy way to answer that. It's from the one carbon metabolism pathways. So you have the one carbon metabolism just affect that. Okay, it's as you know, in animal system, you know that serine hydroxymethyltransferase is involved in controlling cellular methylation and regeneration and biosynthesis of CH3, the methyl group. So just consider that the near isogenic lines are differing in the RHG4 or SN-resistant gene coding for methyl transferases, serine hydroxymethyltransferases on chromosome 8, right? So maybe change in the activity of this enzyme affect the biosynthesis of the methyl group and affect cellular methylation. This is from what comes. This one thing. The second, stress induce, de novo DNA methylation, but it seems like it's directed, it's not random. When you can see it in several papers about droughts, phosphate starvation, all of stuff, you can see DNA methylation affect gene. It's in fact involved in stress response. You can see in response to bacteria, based on stringing, for example, you see that the methylation affect a significant number of genes involved in defense response. So it's not like a general mechanistic response to stress, but it seems like it's targeted for a specific loci and gene involved and related pathways. Is not at random.
You want to get the
Okay.
next question, Juan?
Yes. So there is an anonymous attendee, and this person asked, what statistical tool did you use to locate the specific target fragments that express DNA methylation? Thank you.
Say again.
Changes in DNA methylation.
Say again. I didn't get it. I don't know. So I can.
What statistical tool did you use to locate the specific target fragment that express DNA methylation? I think they mean like changes in DNA methylation that present changes in. So how did you identify the genomic regions that are differentially methylated?
Okay. How did I find that? Okay, so we generated bisulfate reads, whole genome bisulfate reads. Okay, we extracted DNA, we treated with bisulfate, sodium bisulfate, and the make a library, and send for sequencing, and then so we know that methylated cytosine, it's when you treat them with sodium bisulfate, is completely resistant to any conversion, right? But non-methylated cytosine, once you treat it with bisulfate, it can be converted to uracil, right? So with that, you will be able to see which one is methylated and which one is not, okay? And then we identify the location of each single cytosine in the genome. And once we get that, we map this differential methylated cytosine showing increased DNA methylation or reduced DNA methylation compared to the control. And we map them to the soybean genome. Where genome, we mapped them to all the 800 annotated soybean microRNA genes. We know the location, the genomic coordinates of each of these microRNA, the 800, so about 800, right? Annotated. And then we divided the promoter to a 500 base pair region, non-overlapping region, okay? A window. So this 500, another 500, another 500, another 500. And then we compare the DNA methylation in the 500 overlapping window between infected and non-infected samples. And with percentage, we do some statistical analysis, and the methylation differences at least 25%. We'll be able to see this. There's this region are showing significant difference in DNA methylation between infected or non-infected.
Yeah, great.
Okay. Hopefully, this answers this question.
I think so. So our next question is from Jean-Sébastien Parent, who asks, were there microRNAs that changed in abundance between infected and non-infected? I think really you were looking at between susceptible and resistant cultivars, but did not show a difference in DNA methylation. But I think you started by searching for DNA methylation differences and then looked at abundance. So there, I think there could easily be.
Yes, and also maybe I didn't mention this here, maybe this can answer this question. We studied the abundance of the microRNA under non-infected conditions, in the lines, in the susceptible line and the resistant line, without, to see, the basic levels, okay, under non-infected condition. And we found the resistant line, in fact, showing a little bit higher level of microRNA abundance compared to susceptible. This indicates that, in fact, they are, involved in resistance.
Okay. And there is afterwards another question from Banani Mondal, and they ask, do you think these microRNAs can also play a role in root knot nematode infection?
Okay, this is an interesting question. So why not? We didn't test it, but we need to test against different races, different HG type of cyst nematode, right? So we have several races, at least, I don't know how many, but many of races. We tested against only one race so far, HG type 3 or race 3. I'm not sure if it is against race 2 or not, but first we have to test against different races of soybean cyst nematode to see this is resistance. It's against all non-races or not, and then we'll see if this resistance can be confirmed against root knot nematode. In fact, we are thinking about it, but we'll do step by step. Just move test against different races of soybean cyst nematode and then go for root knot. Maybe will have an effect, but most likely will not a huge effect like we saw in soybean cyst nematode. There's some similarity in infection and life cycle between cyst nematode and root knot, but there's a lot of difference as well. And based on my experience, maybe we can see an effect, but I was not expecting to be like huge, like in response to the dots.
So my question is going to take it even a little bit further. My interest, of course, is in the beneficial interactions with rhizobium that lead to nitrogen fixing nodules in the root. And we recently had a lovely review article by Ulrika Matthias on looking at the interaction. So this was in our focus issue in May, looking at how plants simultaneously engage with pathogenic and symbiotic microorganisms. And so she's finding some interactions there. So I'm, of course, very curious whether those microRNAs or maybe their targets are also playing a role in the formation of symbiotic root nodules, which are, of course, another huge carbon sink, just like
that's a very good question.
nematodes.
So this is really very good question, but I will tell you that we already did the same analysis during nodulations. Okay, and we published our paper in Plant Journal, and then we studied DNA methylation of microRNA during nodal development, and found this microRNA also subjected to DNA methylation change during nodal formation during 3 different stages, and we published a paper, but it was very descriptive, and we are now the overexpressing set of microRNA that are differentially methylated to see they are affecting the size, the efficiency of nodulation and nitrogen fixation. So I'm doing this now.
Fascinating. Fascinating. I'm going to have to go follow those up. That's really interesting now to see that connection with the nematodes and the microRNAs and beneficial. Wonderful.
And the good things that we found some common microRNA. If you compare the list of differentially methylated microRNA, the 82 plus the 16 microRNA minus 4, comparing with our list of differentially methylated microRNA during nodal development, there is an overlap.
Of course, there is.
Yeah.
So interesting to see how these things that we think are so completely different, right? Pathogens, symbionts, but of course, it's really all a continuum, and a lot of the things that a microbe has to do to survive inside the plant and to attract the carbon resources that it needs are going to be shared. Right? Because there's only so many ways to get it done. So really interesting. So I'm going to move on to what will be our last question by Pawan Kumar, who asks, do you find any metabolic pathway affected by these 4 microRNAs in susceptible and resistance lines? And you touched on that, but maybe you could just answer that a little more fully.
Okay, so for this microRNA, we have the polyamine pathway. So amine oxidases, it's one of the 2 this microRNA, one of these microRNA targets 2 or 3 gene coding for amine oxidase, and there's a key enzyme involving polyamine metabolism. So I think one of the key pathway that are metabolic pathway controlled by this 4 microRNA, it's a polyamine metabolism. The other gene, it's and also primary metabolic pathway like sucrose. And so it's a sucrose metabolism and polyamine. This is a key to the 2 key metabolic pathway controlled by the microRNA that we functionally characterized.
Interesting, interesting. That was this polyamine pathway was also something that we saw come up in my own research looking at salt and rhizobium, salt stress coincident with rhizobium. So I think that polyamine pathway is pretty important for a lot of interactions, both with biotic and abiotic stress. Okay, so we're going to
very last question.
Yeah, go ahead.
In general, and it's like, what do you think about? Do you think it would be possible, even would be interesting, for instance, to study microRNAs that are differentially expressed in resistant and susceptible varieties to specific diseases as a tool for breeding, for instance, for identifying susceptibility genes, like what you did?
Yeah, I think there is a lot of opportunity to develop like what we call an epigenetic signature. And you can identify epigenetic signature that can define different stage of infection. That's epigenetic, that like the early stage of infection, intermediate stage, later stage, and this epigenetic can be used from it's a signature. It can be determined in the susceptible line and resistant line, and also it can be integrated in a breeding program. So it's not necessary, to select our lines and high performing lines, not only for the resistance, maybe for high yield and quality, okay, based on epigenetic selections and epigenetic signature, and so it's not only using a molecular marker technique and the SNP and analysis, but also you can integrate epigenetic signature if you have some common pattern of DNA methylation chains, hypermethylation, hypomethylation in a specific region. You can use that, and you can do easy PCR, and you can follow it. And in your line during the selection, during your selection or during your breeding program. I think this will be the future, developing epigenetic signature in breeding programs.
Yeah, and on the other side, it would be very interesting to ask a very fundamental question of what's the difference in both plants, why in some plants specific microRNAs are hypermethylated, while in the other plants are hypomethylated. Like, what's the molecular mechanism behind this, right? At the same time, a fundamental scientific question that would be interesting to answer.
Yes, so I think it will be important to see what is the upstream components, not only downstream component. What is triggers this? So, and most likely we are studying this as well from different angles. So it's most likely it's mediated by effector protein, nematode effector protein. We saw maybe nematode effectors secrete some effector that involved in modulating, or modifying the plant methylome or epigenome in general. We have a paper published in Plant Cell in 2018, I think. And we found some nematode effector that target histone deacetylase. And now we are characterizing some nematode effector in tomatoes, and we found they target different component of epigenetic pathways, the methylation, histone modification, small RNA component. So most likely answering your question is characterizing nematode effectors and see how they can, this effector once secreted in the host plant can directly or indirectly, modulate this DNA methylation and histone modification pathways. as well as maybe small RNA component pathways.
So what do you think that the last question, this roughly 8% of difference in the genetic content of both plants, the susceptible and the resistant, would answer for why in one plant is hypermethylated, for instance, one microRNA, and in the other one is hypomethylated in response to the infection?
I think it's mediated through two different things. I think, could be, and it's this nematode effector that's, affecting, modulating different, signaling component, in the resistant susceptible. Okay, so, and then triggering different cascade of signaling, gene expression change, including gene involved in DNA methylations. Well, this could be directly or indirectly.
Okay, thank you.
Yeah, this has been a great conversation, fascinating discussion. I think that epigenetics is clearly something we need to be thinking about. It's an exciting area, and it's something we need to be thinking about now, as you're pointing out, in terms of breeding as well. So we can already start making these links between plant microbe interactions at a mechanistic level, and how we might be able to manipulate that in the plants that we actually cultivate.
Yeah, I just would like to mention something. Sometimes there is not a lot of genetic differences. There is not a lot of genetic, polymorphism, but we can generate the, epigenetic molecules. We can induce and generate epigenetic change, and because the epigenetic modification are stable and can be inherited for many generations, we can use this. So when you have like lines or a species when there is not a lot of genetic differences, this can be very interesting to induce epigenetic change, which can be still inherited to many generations and associated with specific traits.
Yeah.
Okay, well, yeah, thank you. This has been a wonderful what's new in MPMI. And I just would like to remind people to direct them back to Tarek Hewezi's wonderful paper in MPMI that came out in 2020. And we have another installment perhaps coming up soon from him. And so that will be interesting to follow. So anyway, I hope you will, if you return to the page where you registered for this seminar, we should have the recording up in about 24 hours. And by the week, we hope to have a volunteer. Remember, if you are interested in volunteering to help edit the transcript to make it scientifically accurate, please let us know, and we will gratefully accept your assistance. And thanks also to the community then for making this more accessible. Okay, thanks everybody, and until next time.
Thank you, everybody.